Okay, thank you very much, and thank you for the organizer for the opportunity of presenting my work. I'm going to talk about outcome-guided biclustering, a work that I've been doing jointly with Paul Kirk and Chris Wallace. So what I'm going to do, I'm going to present a bit about biclustering. I'm going to talk about the spike and slab lasso or SSLE model that is the starting point of our method. Then, how we introduce more information to better guide the biclustering task using outcome-guided or OGSSLV method, which is the method I propose. And then how we combine many runs to deliver or to output more stable outputs in our OGSSLV model. So let's begin. So biclustering started, let's say, because traditional clustering assumed that we group different samples, and this group of samples behave similarly across all the features or all the genes, if we talk about gene expression data. And this is where biclustering started to become more interesting, because the aim is to group samples across an interesting group of genes and not grouping only samples across all the genes. So in this example, we have these three interesting structures that we want to recover. And in particular, among the many approaches, what we chose is the matrix factorization approach, in which the gene expression data can be computed as a matrix multiplication of lambda times z. Lambda is the contribution to the gene expression data from genes, and z the contribution to the gene expression data from samples. So we want to recover this structure, we just perform this matrix multiplication, and so on and so forth. We can also use this representation, x equals as a matrix multiplication of capital lambda, which, well, takes all the contributions of the lambdas into one matrix, times capital Z transpose. And we can use this factor analysis approach, which is the main approach that SSLV use. The SSLV model, the spike and slab lasso model, is an unsupervised model. It just has the gene expression data as an input. And, well, among all the elements that this Bayesian model-based approach has, it presents the Gaussian likelihood, the data fidelity term, and some priors imposed to the elements of the latent variables. Spike and slab prior to the variable selection samples and genes, if we talk about Z and capital lambda respectively. And then also there's sample membership indicators, some binary matrices, gamma tilde and gamma, with their corresponding priors to estimate these latent structures. What is done is a maximum a posteriori estimate via an expectation maximization approach, and the outputs, the latent structures that we are interested in estimate for downstream analysis. So despite the fact that this is a state-of-the-art model, it has some challenges. It still doesn't recover all the interesting structures that we are interested in, and this is when OGSSLV comes in effect. This outcome-guided structure is just extending the SSLV model by incorporating an outcome variable y, which is in this case the disease presence or absence. And in this model, we assume that y and z are conditionally independent, given the sample membership profile gamma tilde. This allows us to express the joint probability on these factors, and then, this joint probability to be factored as this expression, and then we can model the disease outcome. In this case, what we do is adopt a multi-monomial logistic regression approach. So compared to SSLV, OGSSLV has the gene expression data and also the disease status of samples in a matrix capital Y. You can imagine that it's a binary matrix that tells you which sample has a particular disease or which sample is healthy, and so on. So the structure is similar as SSLV, but we do is incorporate a layer of multinomial logistic regression to estimate the weights and to also guide the biclustering process. These weights will tell us which bicluster is more related to which disease. And the outputs, well, the estimation, again, maximum positive estimate, the expectation maximization, and the output, the latent variables of interest plus this matrix of weights in this case. So this is what we have. We have run many simulations and we have seen this improvement in simulations, but for the interest of this talk, I'm going to talk about directly to real study data. So what we did is take this large gene expression atlas from the University of Tokyo called IMMUNEX-UT. That has 10 immune-mediated disease samples and healthy volunteer samples from a different number of peripheral blood mononuclear cells. And to center our analysis in a very well-studied relationship, we just studied how SSLV and OGSSLV were able to capture the association between interferon signatures and SLE, a very well-studied relation between these signature genes and this disease. Among all the cell types that this dataset has, we'll just focus on monocytes. We are also going to impose something that we call the sparse filtering condition. So among all the biclusters that we're going to estimate, we're going to just focus in biclusters with less than 50% of samples and biclusters that also has more than 10% from a list of curated interferon signature genes, which in this case were 51. As preprocessing steps, standard steps, some batch correction, removing low count genes, and applying some median duration normalization method for our application of SSLV and OGSSLV. So after this normalization, we have a data set with these dimensions, and we run 20 different replicates of both SSLV and OGSSLV. And these are the results. So we found that SSLV were able to capture this well-known relationship in only 7 out of 20 runs. And compared to OGSSLV, which were able to capture in almost every run, 18 out of 20 runs. We also see that OGSSLV, the bicluster from OGSSLV, were more enriched from SLE diseases compared to SSLV. And we can also see this relationship in the distribution of percent of patients of SLE, in this case, in the biclusters. So, yes, OGSSLV is doing a much better work. However, from these 20 replicates, we asked, well, how do we combine all these 20 runs to present a more stable and a much better result from our method? And this is where consensus OGSSLV started also in our work. This is work in progress. To compute this, what we did is standard steps for construct this ensemble approach. We first build similarity matrices between all the biclusters from all the runs using this measure, which is called the Jaeger index. It's an accuracy metric, 1 perfect similarity, 0 non-similarity. Then we apply identical clustering in the distant matrix, and then we cut the dendrogram to get these M groups of similar metaclusters. We compute then the probability of each sample and gene belong to each of these metaclusters, and this probability allows us to assign a sample and a gene to the final consensus bicluster, for which the probability that we are computing here is greater than some threshold. And we found on our experiments, using some grid search approach, optimal probability value for choosing these samples and genes. So yeah, to test this consensus OGSSLE approach that we're doing, what we did is these three steps. First, well, we just combine our 20 runs of OGSSLV following the steps that I explained in the previous slide. Then we identify disease associated by cluster. We just run Fisher's test. And between IMDs, immune-mediated diseases, and healthy samples. And the ones that were significant, and FTR adjusted p-value of less than 0.05, were labeled as candidates of disease modules. We found from all the runs that we found 16 consensus by clusters. And the non-significant ones from FTR adjusted p-value greater than 0.1. Well, you can imagine that it's a very large list. We just took the top 20 of these for a control. We just, as a last step, we validate our findings using open target genetics. We just got a curated list of high confidence genes from these three immune-mediated diseases: rheumatoid arthritis, systemic lupus, and systemic sclerosis. We only, from this validation data, we only include genes with strong evidence from genetic studies, the ones that present a GWAS creditable set score of greater than 0.5. And these are the results. We just do enrichment analysis in a few words, and we found that the biclusters that were labeled as significant were highly associated to the genes that we got from this external data, compared to the ones that we labeled as not significant. 5 versus 1 from rheumatoid arthritis, 2 versus 1 from SLE, and 1 versus 0 from systemic sclerosis. So this is evidence that the set of biclusters that we identify as disease associated show a high frequency of enrichment from these non-GWAS genes. So this is what we've done so far. As a conclusion, we show that indeed OGSSLV improves the detection of disease-related bicluster compared with SSLV. And the results that I show, show highly consistent enrichment of disease-specific signals across replicates. So the next steps is develop a stable and accurate approach for consensus gene and sample loadings. What we have done so far is a consensus of genes and samples membership, which genes and which samples can be in this group of metaclusters. But now what we want is also in a stable way of measure the contribution of the genes and samples in our gene expression data. And then, well, beyond that, we want to adapt this ensemble framework to all bicluster matrix factorization approaches, even beyond transcriptomic data. So yeah, this is what I have to present to you. These are the references. The work of OGSSLV is already published in Statistics and Computing. And if you're curious, this is the code where you can find our project. Thank you very much.